In this video, I would like to talk about your vessels. And this is in a very general sense of what are your blood vessels and what are the properties that make the different vessels different. So your main types of vessels, you've got arteries, and these take blood away from the heart. You have veins, and these put blood in to the heart. And you have capillaries. And these are between the arteries and veins. And they're the site of the diffusion of nutrients and other substances. If we were to look at a vessel, we would see something like this. Here you've got your artery in red, and then you've got a little capillary bed here, and the capillaries are really, really small. And then you've got a vein. And because of what the different vessels do, they do have some structural differences. What I want to do first is start with the general anatomy of these vessels. Both the artery and the vein have this area around the outside of them. This is called a tunica externa, and it's the same name on the artery and on the vein. And it's the externalmost layer. There's a tunica media, again, on the artery and on the vein. This is a muscular layer that's on the inside. The externa is more involved in like anchoring the vessel down. And then you've got a tunica intima, and the tunica intima is the innermost layer, and this is kind of a smooth layer that allows the blood to flow. The difference with capillaries in these arteries and veins is that rather than having three layers, the capillaries are solely made of this tunica interna, or intima, and that allows them to be really, really, really thin, which benefits them in terms of diffusion because it makes the molecules have less distance that they have to travel in order to get from point A to point B. So what do the different layers do? The tunica externa is involved in anchoring the vessels to surrounding tissues. The media is a muscle that's able to constrict or relax as needed to change the diameter of the vessel, which actually changes how much blood flows through it. And the intima is mostly just a smooth surface for blood movement. So I'm going to point out some specializations right now of these vessels. The first one is that I'm hoping you can see that the tunica externa on the artery is much thicker than the tunica externa on the vein. This is because your arteries have to withstand a lot more pressure, so this allows them to do that. Now their tunica media is thicker as well for the same reason. It allows them to be able to deal with the pressure coming off of the heart. In veins, they've got a lot less pressure, and this decrease in pressure means that the blood may flow backwards. So veins actually have these really cool little valves in them, and these valves keep the blood from backflowing so that it only goes in one direction because the pressure is quite low. Now sometimes in veins, these valves break down, and if they break down, it's really hard for you to get blood from your feet all the way up to your heart so that it can get oxygenated in the lungs, and then you usually need something like a compression sock to try to help with that blood return back up to your heart. Veins are also less anchored because they deal with less pressure, so they're a lot floppier. And this, if any of you do blood draws, this may be something that you've noticed. Now, Arteries are going to start big at the heart, and they're going to get smaller and smaller and smaller and smaller, and they're actually going to become go from elastic arteries, which are the biggest ones, to muscular arteries, which constrict and dilate, to arterioles, which are the smallest ones, and that's right before you get to the capillaries. And the arterioles are also able to constrict and relax. Veins do the opposite. They start as venules, these really tiny little vessels, and they branch into veins, and then they eventually branch into the vena cava, the great veins. Now, I've already mentioned to you this idea of constriction and dilation, and part of the reason this happens is your body is only able to maintain so much blood flow at one given time. And it turns out that we don't even have enough blood in our system to have blood running through all of our vessels at the same time. So we basically prioritize which vessels need blood at a given moment. So it could be you've just eaten a large meal. That means your digestive tract is going to get a lot of blood, and it's going to be trying to use that blood to get those nutrients around the rest of your body. During that time, your skeletal muscles might not be getting as much blood to them. Before we move on from our arteries and our veins, I would like to talk about some pathologies. One of these is called arteriosclerosis. This occurs when there's a plaque, which leads to a thickening of the tunica intima. Any thickening of that tunica intima is going to make it harder for blood to flow through, and that's going to mean that the area on the other side of that blockage is having a really hard time getting blood. Next pathology I'd like to talk about is aneurysms. In aneurysms, the vessel wall weakens and the vessel starts to bulge. And this on its own can be problematic, but the larger problem is that if that vessel breaks, it's going to result in a stroke. The last thing I'd like to talk about is the different kinds of capillaries that you have. Now, I want you to look at this picture and note that the capillaries are so small that the blood cells are pretty much going single file through them. And you've got three types: continuous, fenestrated, and sinusoid. The continuous are named because the lining is continuous. It's basically held together very tightly, and this means that amino acids, glucose, and ions, really little things like that, can get through the cracks, but anything bigger can't. And in the case of your brain actually has a modified continuous capillary called the blood-brain barrier. And that has even smaller openings, which allows very little to get to your brain without your brain transporting it there. Fenestrated capillaries have little holes in them, and to me they're kind of the strainers of the capillary world because they allow a lot more to get through. And we will see fenestrated capillaries when we talk about your kidneys, because that's where the most notable fenestrated capillary bed is. The last one is sinusoid, and the sinusoids have these large holes in them, and these large holes actually allow blood cells to enter and exit the capillary. So this is going to happen at your bones, where the new blood cells that have been made are going to go into the bloodstream, and at organs like your spleen and liver, which are going to take the sort of dead and dying blood cells out of the picture and dispose of them. The capillaries are very small. They're only going to allow the blood cells to go through there one at a time, but that doesn't mean that they're not doing much. In fact, where the capillaries really succeed is in their numbers. You've got tons and tons and tons and tons and tons of capillaries. And what this means is that they've got a very large cross-sectional area. Cross-sectional area is basically like if we take all of the vessels together and look at the diameters, we've got a handful of the straws. That's the cross-sectional area. It's not the area of the individual vessel. So this large cross-sectional area means that there's a lot of room for diffusion, and it also means that the blood going through each individual capillary is going very slowly. So this is the perfect thing for diffusion because it allows the nutrients to go into and out of the blood very easily at these capillaries, and then they can go through the veins and get to the heart and be pumped to different parts of the body.